So what if you could, you know, trick a cell into acting young again, completely bypassing the need for hormones just by using a specific sequence of DNA extracted from a salmon?
I mean, it sounds like science fiction, right? But it's actually happening.
It really does. Welcome to today's deep dive, everyone. Our mission today is to explore this truly fascinating biological workaround. Specifically, we're looking at how you can wake up dormant cells in estrogen-depleted tissue, but doing it without actually using a single drop of estrogen.
Exactly. And we are focusing this lens on an area that, well, historically gets totally overlooked or it is quietly dismissed in medicine, which is the female urogenital system after menopause.
Yeah, which is a huge issue for so many.
It really is. We are talking about genuine tissue regeneration in an environment that biology has basically signaled to just shut down.
Right. So, okay, let's unpack this. We're going to look at how a highly specific substance, polynucleotides, which are essentially derived from highly purified salmon DNA, can hack the cellular communication of the vaginal mucosa to, well, reverse tissue atrophy.
And this is such a cool topic because our stack of research today pulls from dermatology, urogenital pathology, and cellular pharmacology.
Yeah, it's a really broad stack. But before we get to the whole salmon DNA part, we need to understand the baseline for you listening. Let's look at the actual biological problem here. What is the, standard operating procedure for this tissue before menopause?
Right. So the standard operating procedure relies entirely on a very specific master regulator, and that's estrogen.
Okay.
In a healthy premenopausal state, the female urogenital tract is incredibly dense with estrogen receptors, specifically, alpha and beta receptors.
Right.
And they operate as this primary communication network for the entire microenvironment. When estrogen binds to these receptors, it orchestrates this massive, continuous symphony of cellular activity.
Like it's constantly sending out marching orders.
Exactly. And the main goal is maintaining a healthy, thick vaginal epithelium. We are talking about a robust mucosal layer that is typically 20 to 30 cell layers thick.
20 to 30 cell layers. It's like a fortress that essentially relies on a daily supply drop of estrogen just to stay standing.
That is a perfect way to frame it. That fortress is continuously fortified. But the thing is, when menopause hits, ovarian estrogen production just ceases. The supply drops stop entirely.
And the cellular fallout from that withdrawal has to be pretty dramatic, right?
Oh, it's incredibly dramatic. Without estrogen acting as the master regulator, collagen synthesis plummets, particularly collagen types I and III, which are, the fundamental proteins that give the tissue its strength and elasticity.
Right, and it isn't just a passive decline where the tissue slowly stops building.
Yeah.
The research points out that the degradation actually accelerates, doesn't it?
Yes, it actively breaks down. In a low estrogen environment, the activity of a specific group of enzymes just skyrockets. These are called matrix metalloproteinases, or MMPs.
MMPs.
And their primary job in the body is to break down collagen and elastin fibers. Usually they are kept in check, but without estrogen, they go into total overdrive. So you have a dual front crisis here. The tissue has stopped building new structure, and these enzymes are actively destroying the structure that is already there.
Okay, let's give you, the listener, a way to visualize this. Imagine your tissue is a construction site. The cells responsible for building the fibroblasts are the construction workers, and estrogen is the site foreman.
I love this analogy.
Right. So when the foreman retires, the workers don't know what to do, so they just put down their tools. And to make matters worse, a rogue demolition crew, those MMP enzymes you mentioned, takes over the site and literally starts tearing down the scaffolding.
That captures the mechanical failure perfectly. And for the person actually experiencing this, and statistically a massive portion of our listeners will, or already are, it's not just an interesting biological shift.
It's intensely physical.
Right. The clinical terms are genitourinary syndrome of menopause, or GSM, and vulvovaginal atrophy, VVA.
Which impacts an enormous number of women.
Yeah.
Yet it's something people rarely talk about in anatomical, empathetic detail. Like we're talking about daily physical discomfort, painful intimacy, and just a profound loss of quality of life.
Exactly.
So what is physically happening to that microenvironment?
Well, macroscopically, the tissue literally changes color. It becomes pale because the vascular network is shrinking. It loses its elasticity. The vaginal canal can actually narrow and shorten, and the labia minora can regress.
Just from the lack of estrogen.
Yeah, structurally, because estrogen is no longer there to promote the production of nitric oxide, vasodilation just plummets.
And nitric oxide is what tells blood vessels to relax and open up. Right?
Yeah.
So we were talking about a severe drop in blood flow.
Significantly less blood flow. And I mean, in mucosal tissue, blood flow is everything. Less blood flow means less sensitivity, less natural lubrication, and less engorgement of the erectile bodies. Histological studies, like when you actually look at the tissue under a microscope, they show that nerve signaling becomes completely muted. The actual nerve density is reduced.
So for the patient, this can translate to localized numbness or just requiring far more mechanical stimulation to achieve any sensation at all.
Yeah, the sources mention a complete disruption of sexual function, delayed responses, weaker responses, or just outright pain.
And this extends way beyond sexual function, right? The urinary tract takes a massive hit as well.
It does. The lower urinary tract and the vaginal tissue actually share the same embryological origin, meaning they developed from the exact same primary cells in the womb.
Oh, I see.
Because of that, the urethra and the bladder base suffer the exact same estrogen withdrawal. This leads to what's historically called urethral syndrome.
Which sounds awful.
It is. The urethral lining thins out, the smooth muscle loses its sensitivity, and the natural folds of the urethra just disappear. This translates to intense urinary urgency, waking up constantly at night, and incontinence.
Wow, so it's a cascading failure of the entire local ecosystem.
Exactly.
Which brings us back to the builders on our construction site, right? The fibroblasts. Let's focus on them, because understanding how they behave seems to be the absolute key to this entire deep dive.
It really is. Fibroblasts are incredibly dynamic cells. They aren't just passive bricklayers waiting for orders. They are highly reactive to their environment, specifically to mechanical tension and biochemical stress.
Okay.
When tissue is damaged, or in this case severely atrophied, fibroblasts actually sense that structural weakness. In response, they can transition into a highly activated defensive phase. We call these protomyofibroblasts.
Protomyofibroblasts. It sounds like they were putting on a suit of armor to go to war with the atrophy.
They essentially do. They equip themselves with internal contractile bundles called stress fibers. These are tiny, powerful protein chains that allow the cell to physically grip the surrounding matrix and pull.
So they're trying to hold it all together.
Right. They generate traction forces to pull damaged tissue together, trying to rapidly patch the failing structure.
But the research highlights a massive risk here, right? Because if this localized, panic healing process goes wrong, they aren't building healthy, flexible mucosal tissue.
No, not at all. If the tissue lacks the right organizing signals, which it does because the estrogen is gone, the extracellular matrix they frantically build is wrong, it becomes incredibly stiff. Biologically, it starts to resemble scar tissue.
And scar tissue is functionally useless for a vaginal wall that needs to expand and contract.
Useless and structurally compromised. Scar-like tissue has an overabundance of type 3 collagen. Healthy, elastic mucosal tissue relies heavily on type I collagen.
Ah, I see.
So when you flood the area with type 3, it loses all its compliance. This disrupted fibrotic healing process isn't just uncomfortable, it is directly linked to severe mechanical failures like pelvic organ prolapse.
Wow.
The vaginal fibroblasts essentially recognize the estrogen-depleted thinning tissue as a massive wound. So they panic-build stiff scar tissue, which is brittle and ultimately fails to support the heavy pelvic organs resting above it.
Okay, wait, I have to challenge this premise, though. Because if estrogen is the fundamental life support signal for this specific tissue, the only language these fibroblasts truly understand, how can we possibly expect to just bypass it?
That's a very fair question.
Right, because you can't just throw a random chemical at a cell and expect it to rebuild a highly complex mucosal wall without the primary hormone. If the main communication lines are down, how do we give the fibroblasts the instruction to build healthy tissue again instead of just panic-building scar tissue?
Yeah, that is the million-dollar question in regenerative medicine right there. If the front door, the estrogen receptor, is locked, you have to search the cell for a back door.
A back door.
Biology rarely relies on a single point of failure. In this case, the back door is a completely different set of receptors called adenosine A2A cell surface receptors.
Adenosine A2A receptors. Okay, but why does the body even have these if it uses estrogen to run the show?
Because these receptors are ubiquitous. They are found all over the human body. Evolutionarily, they act as a profound regulatory network, mostly heavily concentrated in immune cells, endothelial cells that line blood vessels, and crucially, dermal and mucosal fibroblasts.
So they're like a backup system.
They're essentially a universal distress and repair frequency. They modulate inflammation and oxygen delivery. If you can figure out how to selectively stimulate the A2A receptor, you can command that fibroblast to heal and rebuild, totally bypassing the need for estrogen entirely.
And this is exactly where the molecular hacking comes into play. Here's where it gets really interesting for you listening. The star of our research stack today is a substance called polynucleotides, often referred to in the literature as PN or PDRN, polydeoxyribonucleotides.
Yes.
And the fascinating part, they are derived from highly purified salmon DNA. We're looking at clinical therapies that literally put fish DNA into human tissue to trigger a localized healing response.
I know, it sounds bizarre at first glance, but the biochemistry is incredibly elegant. PDRN is essentially a long, highly purified chain of nucleotides, which are the basic structural units of DNA. Right.
When this substance is introduced into the atrophic vaginal mucosa, it doesn't just passively float around hoping to do some good. Because of its specific molecular shape and weight, it acts as a highly specialized physical key.
Okay.
It binds directly and selectively to those adenosine A2A receptors sitting on the surface of the dormant fibroblasts.
Let's break down exactly how polynucleotides signal fibroblast activation, specifically in this estrogen-depleted environment. Are we just delivering the raw materials, like dropping off a load of bricks at our construction site, or is this DNA actually giving the builders new blueprints?
What's fascinating here is that it provides both, which is why it is so effective in a depleted microenvironment. Let's start with the bricks. Over time, these long polynucleotide chains are naturally broken down by enzymes in the tissue. As they break down, they release a massive pool of nitrogen bases and nucleosides. The dormant fibroblasts scoop these up through something called the salvage pathway.
The salvage pathway, meaning the cell doesn't have to manufacture these basic building blocks from scratch.
Exactly. Synthesizing DNA and cellular components from raw materials takes an immense amount of cellular energy that atrophic dying tissue simply doesn't have.
Right, because it's starving.
Exactly. By providing these components directly, the salvage pathway saves the cell massive amounts of energy, instantly boosting its viability. But providing raw materials doesn't explain the structured regeneration. The primary driver, the actual blueprint, is the active signaling.
Okay, let's talk about the blueprint. The research stack is full of dense biochemical acronyms like VGF, FGF, MAPK pathways. Instead of just listing the alphabet soup of inflammatory pathways, what is the actual mechanism? How is this salmon DNA changing the internal software of the cell?
Think of the A2A receptor as a master override switch. When the polynucleotide binds to the A2A receptor on the outside of the fibroblast, it flips that switch.
Okay.
This activates an enzyme inside the cell that immediately floods the interior with a molecule called cyclic AMP, or CAMP.
We've talked about CMP before on the show. It's like pulling the fire alarm, but for cellular regeneration instead of evacuation.
That's a great analogy. That flood of CAMP completely rewrites what the cell is currently doing. First, it commands the dormant fibroblasts to rapidly multiply and proliferate.
It wakes them up.
Yeah, and then it initiates the rebuilding of the two things the tissue lost during menopause: blood supply and structural integrity.
Wait, how does it get the blood supply back without the estrogen to trigger it?
The CAMP cascade forces the fibroblast to release growth factors that specifically target blood vessels, primarily VEGF, vascular endothelial growth factor. It initiates angiogenesis, the literal sprouting of brand new capillary networks.
That's incredible.
It is. This directly reverses that pale, blood-starved tissue we talked about earlier, bringing back the oxygen and fluid necessary for sensitivity and lubrication.
Wow.
And simultaneously, it forces the fibroblasts to start synthesizing fresh, highly organized type I collagen and elastin.
Meaning it forces them to build the flexible scaffolding, not the stiff type 3 scar tissue they normally panic build.
Precisely. It organizes the matrix, but remember, we still have that rogue demolition crew roaming the construction site, the MMP enzymes that were tearing everything down.
Right, because if you are building new scaffolding while the demolition crew is still working, you are just running in place. How does the DNA hack address the destruction?
This is perhaps the most brilliant part of targeting the A2A receptor. Activating this specific backdoor simultaneously cuts the power to the demolition crew's tools.
It just shuts them down.
Completely. The chemical alarm bells that normally cause chronic inflammation and tissue breakdown in atrophic tissue are silenced. The A2A receptor suppresses the release of inflammatory cytokines, which are, the chemical messengers that recruit destructive immune cells to the area.
Okay, all right.
And crucially, it completely blocks the production of MMP1.
So it literally fires the demolition crew.
It stops them dead in their tracks. So in this entirely estrogen-depleted environment, simply by introducing these polynucleotide chains to the A2A receptor, you achieve a simultaneous increase in high-quality structural building, the growth of new blood vessels, and a total cessation of tissue degradation.
So for you listening, what does this all mean for the actual treatment of vulvovaginal atrophy? If someone is dealing with the painful realities of VVA, how is this actually applied to the tissue?
Well, the translation from cellular biology to clinical application is found in specific topical and injectable formulations, most commonly referred to in the sources as PNHA.
PNHA, polynucleotides combined with hyaluronic acid.
Yes. They are formulated as ovules, gels, or targeted injectables designed specifically for the vulvovaginal area.
And what is the biological logic of combining the salmon DNA with hyaluronic acid? Like, why not just use the polynucleotides on their own?
It comes down to creating the perfect healing microenvironment. Polynucleotide polymers are highly hydrophilic. They absolutely love water.
Okay.
When they are introduced into the dry, atrophic vulvovaginal tissue, they chemically organize into a three-dimensional gel matrix. This gel binds tightly to ambient water, creating a massive, localized moisturizing effect.
Ah,
When you pair that specific structural geometry with hyaluronic acid, which is globally recognized for its profound hydrating properties, the results are deeply synergistic.
so the patient gets immediate soothing relief from the severe dryness, burning, and mechanical friction associated with the atrophy, simply because of that hydration matrix.
Exactly. The hyaluronic acid acts as the immediate first aid. It restores the physical cushioning. But the polynucleotides are playing the long game. While the hyaluronic acid provides that immediate symptomatic comfort, the polynucleotides are slowly permeating the lamina propria.
The lamina propria being the deeper foundational connective tissue layer beneath the surface.
Right, the basement level of our fortress. Down there, they begin relentlessly binding to the A2A receptors on the fibroblasts. Over the following weeks and months, they drive robust neocollagenesis and neoelastogenesis, the creation of new, youthful collagen and elastin.
They're essentially doing the heavy lifting over time.
Yeah, they stimulate the basal cells to rapidly divide, physically thickening the vaginal wall back toward that healthy 20 to 30 cell layer thickness we see in premenopausal tissue.
They are genuinely changing the macroscopic architecture of the tissue, which has a profound secondary effect on the vaginal microbiome, doesn't it? Because the microbiome relies entirely on the physical structure of the tissue to survive.
Absolutely. The two cannot be separated. In a healthy, thick vaginal epithelium, the superficial cells, the ones right at the surface, are packed with a carbohydrate called glycogen.
Okay.
As the tissue naturally turns over and these cells slow off, the local good bacteria, primarily lactobacilli, feed on that glycogen.
They eat the glycogen and ferment it.
Exactly. They ferment it into lactic acid. This continuous production of lactic acid is what maintains the highly acidic pH of a healthy vagina. That acidic environment is the ultimate defense mechanism. It kills off invading pathogens and prevents painful urinary tract infections and cystitis.
Wow. Okay.
But in an atrophic, thin vagina, there simply aren't enough cell layers to produce a glycogen barrier. The lactobacilli snarve, the pH rises, and chronic infections take hold.
So by using the polynucleotides to force the tissue to thicken and rebuild those 20 to 30 layers, you are restoring the glycogen food source. You're basically resurrecting the natural ecosystem.
You are. If we connect this to the bigger picture, standard lubricants or traditional moisturizers are just symptom masking. They sit on the surface, provide temporary slip, and then they are gone.
They do nothing for the underlying cellular failure.
Nothing. But PNHA formulations are genuinely altering the cellular microenvironment. They are forcing the tissue to behave as if the biological foreman is back on the job, completely restoring the tissue's structural integrity, its elasticity, its vascular blood flow, and its natural pH defense mechanisms.
And the most incredible part of this deep dive is that it is doing all of this without the use of systemic or even local hormonal therapies, which is a massive paradigm shift. Millions of women either cannot take estrogen due to a history of hormone receptor-positive breast or ovarian cancers, or they simply prefer to avoid navigating the complexities and potential risks of hormonal treatments.
It offers a purely regenerative, localized solution to a debilitating problem. For decades, the medical consensus was that this type of atrophy was solvable only by replacing the missing hormone. This research proved that biology is far more adaptable than we gave it credit for.
It's amazing. Let's pull all of this together. Menopause fundamentally removes estrogen, which acts as the tissue's master regulator and construction foreman. Without it, the fibroblast builders stop working, the blood vessels shrink, the nerves go quiet, and the MMP demolition crew starts destroying the vaginal mucosa, leading to structural weakness and daily pain.
Right.
But then polynucleotides step in as a master molecular hacker. Derived from purified fish DNA, they completely bypass the broken estrogen communication lines and bind directly to the ubiquitous A2A backdoor receptors.
Beautifully summarized.
Thank you. This flips the cellular switch back to build and repair mode, elevating internal CANT-MP, triggering new blood vessels, silencing the inflammatory demolition crew, and effectively turning back the biological clock on that local tissue.
It truly is a masterclass in exploiting alternative cellular pathways. It highlights just how much potential is waiting in the wings of our cellular biology, provided we know which molecular buttons to push.
It redefines what we think of as permanent biological decline, which leaves you, listening to this, with a final, slightly mind-bending thought to mull over. We've just spent this deep dive exploring how we can use a specific sequence of DNA to completely bypass hormonal signaling and regenerate severely atrophic tissue, simply by targeting this one ubiquitous A2A receptor.
Yeah.
If we can achieve genuine structural and vascular regeneration here, could this exact same backdoor communication be the key to reversing age-related decay in other organs that rely heavily on hormones? If we can literally trick a localized cell into thinking it's young again, commanding it to rebuild blood vessels and collagen without a single drop of systemic hormones, what are the true biological limits of human tissue regeneration?
That is the question.
Something to think about the next time you hear someone say a condition is just a natural part of aging. Until next time.